Welcome back to The Explainer. Today, we are jumping straight into something that is literally a modern medical marvel. We're going to completely demystify the science of vaccines. But we're not just going to skim the surface here. We are going to trace the incredibly fascinating connections between microscopic cellular biology, the macroscopic math of global health, and the physical engineering that makes this whole shebang possible. So, grab your notebooks, because we've got a brilliant journey ahead of us today.
Here's our roadmap. We're going to build our knowledge step by step, starting with the biology of immune memory, then calculating efficacy, the base rate fallacy, herd immunity and R0, vaccine technologies, and finally, delivery and cold chains.
Okay, let's dive right into it. Section 1. The biology of immune memory, our cellular blueprint.
At the very heart of all vaccine science is this exact concept: immunological memory. This is the ultimate goal of any vaccination, really. It's the remarkable ability of your immune system to quickly and very specifically recognize an antigen, which is just a foreign substance, that your body has encountered before. Once it recognizes it, it kicks off a highly targeted immune response. It's honestly a lot like a microscopic wanted poster that your cells just keep on file for the future.
So let's look at how this memory actually plays out. On the left side of this graph, which represents your very first infection, you can see a curve that takes time to build up, peaks, and then drops off. But follow that timeline over to the right, years later, to a mild reinfection. Just look at that massive, rapid, near-vertical spike in antibodies. No hesitation at all. That is immunological memory. A vaccine basically safely gives your body that first learning curve, practically guaranteeing that massive second spike if you ever face the real pathogen.
Moving right along to Section 2. Calculating vaccine efficacy, trials, and data.
Now, how do we actually measure this memory out in the real world? Well, to calculate efficacy under controlled conditions, scientists run massive clinical trials. We're talking tens of thousands of people. These participants are split right down the middle. Half go into a placebo group, meaning they receive no active vaccine, and the other half go into the vaccinated group. Then we simply wait and monitor who gets sick over time. By directly comparing the illness rates between these two groups, we can isolate exactly how much protection the vaccine is actually providing.
Let's put some real numbers to this. In this data from a Pfizer/BioNTech clinical trial, out of 43,000 total participants, 170 people eventually became infected. But notice the distribution here. 162 of those sick individuals were in the placebo group, while only 8 were in the vaccine group. That is a massive disparity. And that's exactly what gives us our efficacy rate. Now, the absolutely crucial point here is understanding what 95% efficacy actually means. It doesn't mean that if you expose 100 vaccinated people, 5 of them are just automatically going to fall ill. No way. It actually means that each vaccinated individual is drastically less likely to get sick compared to an unvaccinated person standing in the exact same environment.
Which brings us to Section 3, the base rate fallacy, deconstructing the math.
You might have heard a statement on the news like this: the majority of people who got the disease were actually vaccinated. Now that sounds super alarming, right? Like the vaccine isn't working at all. But let's set up a hypothetical outbreak scenario using these figures to visually anchor the math. We're going to see why that statement is actually a classic statistical illusion called the base rate fallacy.
Let's patiently deconstruct this. Imagine a population of 500 people exposed to a rare disease outbreak. In this highly immunized community, 490 people are vaccinated and only 10 are unvaccinated. Now, let's say the disease spreads and 10 vaccinated people fall ill and 10 unvaccinated people fall ill. If you just look at the raw numbers of sick people, it's a 50-50 split, 10 of each. You'd naturally think, wow, the vaccine did nothing. But wait, look at the base rates. Those 10 sick unvaccinated people, they represent 100% of the entire unvaccinated population. Every single one of them got sick. But the 10 sick vaccinated people, they represent just roughly 2% of the 490 vaccinated individuals. The crucial takeaway here is that the percentage of vaccinated individuals who get sick will always be drastically lower. When a population is heavily vaccinated, the starting pool is just much, much larger.
Let's zoom out even further and look at Section 4, herd immunity, and R0, protecting the community.
Imagine a community with absolutely zero immunization. The infectious agent just passes freely, rapidly, and chaotically from contagious individuals, in red, directly to the susceptible ones, in blue. There are absolutely no barriers. It just runs wild. But look what happens when we introduce immunized individuals into the mix, represented here in yellow. Notice how they literally act as a physical wall. The contagion just cannot freely pass through them. Because the virus hits a dead end, the susceptible individuals on the other side are indirectly protected. And that right there is the definition of herd immunity. When a large enough proportion of a community becomes immune, it protects both the vaccinated and those who simply cannot be vaccinated.
So how many of those yellow figures do we actually need to build that wall? Well, it all depends on a number called R0, the basic reproduction number, which basically measures contagion. Check out this table. Smallpox has an R0 of 3.5 to 6, meaning one sick person infects about 4 to 6 others. Measles, on the other hand, has a massive R0 of 12 to 18. It is insanely contagious. The rule of thumb here is simple. The more contagious a disease is, the higher the percentage of individuals who must be immune to achieve herd immunity and stop the spread. So measles demands a significantly denser wall of immunized people than smallpox does.
All right, on to Section 5. Types of vaccine technologies, the mechanics.
Okay, this flowchart might look like a giant puzzle, but let me be your tour guide here. Starting right in the center, with a virulent parental virus, scientists have a bunch of different engineering paths. They can use attenuation, which weakens the virus so it replicates but doesn't actually make you sick. This is how the live polio vaccine works, which, fun fact, uses strands like P3 Leon isolated all the way back in 1937 from an 11-year-old boy in Los Angeles. Or they can go the subviral route down on the bottom left, creating a virus-like particle, or VLP. The HPV vaccine uses this trick. It takes a single viral protein that self-assembles into an empty shell. It looks exactly like the virus to your immune system, but there is absolutely zero genetic material inside.
Another major path is inactivated or killed vaccines. For this, scientists use chemical procedures like formaldehyde or physical procedures to completely eliminate the virus's ability to infect. It is dead. However, it still looks like the threat to your body. The massive advantage here is safety. It literally cannot cause the disease, but this introduces a unique engineering problem. Because it's dead, it doesn't replicate, which results in lower immunogenicity. It's kinda quiet and doesn't trigger the immune system as aggressively, which is exactly why these types of vaccines often require multiple booster shots.
And this is where scientists got really clever with a biological workaround: adjuvants. An adjuvant is just a compound added to a vaccine that specifically stimulates and boosts the immune response. Since those inactivated vaccines are a bit quiet, scientists add things like aluminum salts, commonly known as alum, or oil and water emulsifiers. These adjuvants act like a biological alarm bell. They induce innate immunity and activate inflammation, basically forcing the body to pay very close attention to the vaccine so it forms that crucial long-term memory.
And moving over to the absolute cutting edge of vaccine mechanics, we have nucleic acid-based approaches. Compare the Johnson & Johnson technology with Pfizer and Moderna. Both are remarkably similar in what they're trying to do. They don't deliver the virus itself, but rather the genetic blueprint for the antigen, specifically the spike sequence. Where they actually differ is the delivery vehicle. Johnson & Johnson uses an adenovirus capsid, which is just a harmless modified virus shell, to smuggle the sequence into the cell. Pfizer and Moderna use a lipid nanoparticle, basically a tiny microscopic bubble of fat, to deliver the mRNA blueprint. Different vehicles, same destination, teaching our own cells to print out the wanted posters themselves.
Finally, Section 6, Delivery and Cold Chains, Engineering the Real World.
An often completely overlooked but absolutely critical part of global health is the cold chain. We've got these amazing biological blueprints like lipid nanoparticles and live-attenuated viruses, but they are incredibly fragile. If they get even a little too warm, they degrade and become useless. Transporting them across the globe requires a flawless, unbroken chain of high-tech coolers and specialized refrigeration units. Getting it from a state-of-the-art manufacturing plant all the way to a tiny rural clinic at the end of a dirt road is a massive logistical and engineering triumph.
So what is next for delivery engineering? Well, check out this tiny little square resting on the fingertip here. That is a microneedle patch. Traditionally, we do vaccines via injection, orally, or maybe a nasal spray. But these patches contain microscopic needles made of dissolving sugars or polymers mixed right in with the vaccine. You just press it onto the skin. The microneedles painlessly dissolve, and the vaccine is delivered straight into the immune-rich layers of your skin. It completely gets rid of sharp needles, and best of all, often reduces that strict reliance on the complex cold chain we just talked about. It's a real game changer.
We have covered some serious ground today.
We've seen how a microscopic cellular memory actually scales up to create a macroscopic shield for our entire community. And we've seen how ingenious chemical and mechanical engineering takes it from a lab concept to a global reality. Which leaves us with this final, exciting thought. As our vaccine platforms continue to evolve, moving from whole, weakened viruses in the 1930s to precisely coded lipid nanoparticles today, what new diseases will this microscopic engineering allow us to conquer next? HIV, malaria, maybe even cancer. The blueprint is in our hands. Thank you so much for joining me today. Keep learning, and I will see you on the next explainer.